So let's begin. What did we kind of end with last time? We started in the wonderful, awesome world of what? Cells, right? We started talking about these specific structures in eukaryotic cells called what? Begins with O. Organelles, all right? So we're going to continue that discussion. Now we're going to continue that with the organelle. We ended with the nucleus, right? Now we're going to pick up with another organelle called the endoplasmic reticulum. And this is actually more of an umbrella term for two organelles, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So the endoplasmic reticulum is a series of organelles. Typically you find this around the nucleus. Okay. Now, what are the two types? Smooth and rough, right? So, rough ER, we're going to find this close to the nucleus. Okay, smooth, not so close as the rough endoplasmic reticulum. So what does the rough endoplasmic reticulum do? Well, its location kind of is inherent or important in its function. So what does the rough ER do? Does anyone know? What macromolecule pops into mind when you think of the rough endoplasmic reticulum? Protein. Why do we call it a rough endoplasmic reticulum? Ribosomes. It has ribosomes. It's studded with ribosomes. What do we use ribosomes for? What do ribosomes make for us? Protein. They're going to make protein. What goes into the ribosome? What goes into the ribosome? What macromolecule? What molecule? Guesses? What? You're banging on the door of being correct. Close. Nucleotides and a long string and a single strand. Nucleic acids are mRNA, messenger RNA, correct. So you were knocking on the door being correct on that. All right, so let's talk about the rough ER here. The rough ER is a folded membrane very close to the nucleus. And we have these rough points, and that's ribosomes. Okay, so we have our nucleus here. There's my chalk on my finger. We have our iPad. You're going to get thrown on the wall. Okay, never mind. Oh, well, thank you for working. I need to draw this out on here so people can see it here. Okay, so the rough ER has the ribosomes on it, and ribosomes are for protein synthesis. Expletive, not worried about it. Okay, rough ER. Here we have, what is in the center of our nucleus? What do we have? Our DNA, right? DNA makes RNA. RNA leads to protein, right? So here is our, I'll draw it up here. Here is our nucleus up here, and we make what from it? RNA, right? RNA then leaves the nucleus. RNA, messenger RNA to be specific. We've got a long strand of nucleotides, right? And this carries a copy of the information of DNA from the nucleus. Now we have our rough endoplasmic reticulum, but I'm going to draw a side view of the rough ER. So here we have this open organelle, and we have these rough points, which are our ribosomes, okay? Now in the diagram on here, do you see the little purple dots on that? Those are the ribosomes I'm drawing up here. Now the messenger RNA is going to interact with one of these ribosomes. The ribosomes are going to read our mRNA here, and as it reads that mRNA, what will it do? It makes protein. And it's going to make that protein, is going to be shunted into our rough endoplasmic reticulum. So messenger RNA interacts with the ribosomes on the surface of our rough ER, and it produces protein inside of our rough endoplasmic reticulum. Okay? Once it gets in there, you're going to have some modifications. You're going to have, for example, a signal peptide cutoff. You're going to have some things happen to that protein as it enters into our cell, as it enters into our rough ER. Okay? Now, why does it make sense that our rough ER is very close to our nucleus? Exactly. When we start talking about structures of RNA, we're going to see that RNA is an ephemeral molecule. It doesn't last a long time. Okay, it's not a very long-lasting structure. It's going to break down. It's very unstable when you compare it to DNA, for example. Now, I want to make another point is, I don't want to leave you with the idea that all protein synthesis happens with the rough ER. Protein synthesis is going to happen elsewhere, but this is one location for protein synthesis, not the only location for protein synthesis. Okay. Any questions on that? Okay.
Yeah,
Next. Yes.
On the test, we're going to need to know that model, that DNA linked RNA, which makes the mRNA that goes into.
No, you won't have to draw that model out. You won't have to identify that, but I could ask a question about it. Like, for example, okay, let me say this. I wouldn't ask anything other than what I've talked about in the slides. Okay, you don't have to know this model per se, but you should have an idea of what happens. mRNA interacts with the ribosomes on the rough ER. The ribosomes then make protein that is shunted into the rough endoplasmic reticulum. Okay. Okay, and the next one is the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is important in, well, several areas. We won't get to it now, but it's also important in muscle physiology. Now, smooth ER, why is it smooth? There's no ribosomes, correct. It does not synthesize proteins like you would think that the rough ER does. All right. Important here is it's going to make lipids, fatty acids, those sterile compounds we talked about before. And it's also important in detoxification. For example, you're going to see a lot of smooth ER in the liver, for instance.
So it's not just like in one spot.
Well, let me say this. No, it's going to be in all your cells, but it's going to be okay. Let me step back a second. Do you remember we said that cells, when we showed that picture, that's a generalized picture? Okay, different cells will have different amounts of smooth ER depending on their function. Okay, so some cells will have more if that's inherent to their function. Some cells will have less if that's connected to their function. So it just depends on the cell's function. Okay. So let's talk about this detoxification effect. When you think of detoxification, what do you think of? Breaking down alcohol and drugs, right? If someone is an abuser of drugs or alcohol, what do you usually think of? If someone drinks a lot, does it take more alcohol to get them drunk or less? Less, more. It takes more. It takes more alcohol. If someone's never drunk before and they start drinking, and then someone is a raging alcoholic or a raging drug user, does it take more drugs to get them high and to get drunk or less? It's more. Okay? The reason is your body is doing detoxification, okay, connected to the smooth ER. As you drink more, use drugs more, your body's detoxifying faster. It becomes more efficient at it. Okay? Now, what happens if you are an abuser of alcohol? What's a problem that happens? Your liver, exactly. And we start looking at where you see a lot of smooth ER, you're going to say the liver is one of those areas. So it does have ramifications. Okay. Any questions so far?
So where there needs to be more detoxification, there'd be more smooth ER than there would be rough.
Well, not so much a ratio. The smooth ER would be more efficient at making the enzymes necessary for it. Okay. Next is the Golgi body. Golgi body. Now, when you think of now, this is another organelle. We're done with smooth and rough ER. Next one is the Golgi body. When you think of that, what do you think of when you say your Golgi body? What comes to mind? What things come to mind when you think of the Golgi body? Carrying, okay, that's a good way to think about it. A transport system, right? Okay, it's really important in transport, but it's also a little bit more than transport. It's a transport and modification system. Modification is in when things come in, there's going to be a few modifications to what comes in. When it transports it, it's going to release it with those modifications. A little bit else than that is you go to UPS, right? You ship something via UPS. What does UPS do to that product? It directs it to a certain place, right? Okay. Well, it's similar to that direction because it's going to direct your products to certain areas. For example, some proteins may need to go outside of the cell. Some proteins may need to stay inside the cell. Some proteins may need to go to the membrane of a cell. It just depends on what you're doing. And the Golgi body will direct certain proteins to certain areas. And we'll give some examples in a minute.
Do all proteins go through that?
No. No, many do, especially from the rough ER, but not all proteins will go through the Golgi body. Okay. So, couple function, it will receive and modify proteins from the rough endoplasmic reticulum. Next, it will package them up. It will process those. And then third, it will release them via secretory vesicles. And we'll talk about each one of those coming up here. Okay, any questions? We're going to go through each one of these steps. Okay, we're going to show each one of these working. Okay, so function 1 is we receive proteins from the rough endoplasmic reticulum. On the left right here, we have our rough endoplasmic reticulum right here. Let me just draw it here. This is our rough endoplasmic reticulum. You see that? That has produced our proteins we drew on the board. Next, that rough endoplasmic reticulum will release those in vesicles, which are little membrane pouches, if you will, and that transports it over to our Golgi body. So here's our protein inside here, and it transports that protein to our Golgi body. Right? So next, what's going to happen? It's going to do what? It's going to enter inside the Golgi body, right? The protein will enter inside the Golgi body, and that happens by a fusion process. That membrane fuses with the exterior of the Golgi body. So let me kind of draw that out here really fast here. We'll talk more about this when we talk about membranes and how they function. But we have the membrane of the Golgi body here. It's that bilayer membrane. Here is our vesicle here. And this will fuse with the membrane. And as it fuses, what's going to happen is this part of the membrane will actually become part of the membrane of our Golgi body, like that. See what's happening? These two connect, and they're made of the same thing, phospholipids. And as it combines, it actually connects and becomes one. Now, if we have proteins in here, guess what those proteins are going to do? Go into the Golgi body. Okay, endocytosis. And we'll talk more about this when we talk about the models of fluid dynamic models of the membrane. Okay. Next, we got the proteins inside of our Golgi body. Then what are we going to do with them? We're going to move them around. Exactly. We're going to move them around. We're going to process them. What do I mean by processing them? We can add we can add carbohydrates to them. We can make sure that they are in their correct shape. Remember, shape's the thing with proteins. Shape is inherent in the function of a protein. Then what are we going to do with them? We've done shaping them. We've put carbohydrates on. We've done all the stuff to them. Then what? Send them where they're going to go. Exactly. We're going to do that final sorting at the end, and we're going to send them off along their way. So what are some options here? What can we do with these things? First, we can put them in we'll put them in these vesicles, and one, we can release it outside the cell. So for example, this one right here, number 7 on this diagram, here's the secretory vesicle. It fuses with the outer membrane. See how the outer membrane makes this fold inside? See that? Exocytosis. It fuses with the membrane, just like we drew up here a second ago, and it releases those proteins outside of the cell. Exocytosis. How about another one? So that's one way. How about another way? Another option is, what if we need to add proteins to the outer membrane, like receptors? There's certain things. Remember we said that the cell will hear things about its external environment through receptors on the membrane? Remember that? Okay. We can actually put these membranes inside the vesicle. Do you see how these membranes are going across the vesicle like that? See that? Up there, you see that? Where it's on both sides of the membrane of that vesicle? When that vesicle fuses with the membrane here, that membrane integrates into the outer membrane, just like we drew up here. What's that going to do? If we have proteins in that membrane of the vesicle, and that vesicle fuses with the outer membrane, what's going to happen? Exactly. It's going to transfer because those proteins are part of that vesicle. Okay, let me just draw it real fast. Okay, so here, I'll just even use the same image here. So here we have our vesicle, which is basically a little ball of bilayer membrane ball. Okay, and we have our proteins going here, 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 and here. You with me? This vesicle fuses with the membrane here. Again, same thing. It integrates into the membrane like that. As it integrates, it will straighten out, and guess what happens? This is the outside of the cell, and inside. We're going to introduce these proteins across the membrane. So we can introduce proteins on the outer membrane of the cell by putting it on a vesicle. The vesicle fuses with the outer membrane. We've got that integration. What's that?
So as proteins go into the cell, would it change the DNA and the RNA of the cell?
Loaded question on that. And why is it loaded? You mean like cause mutation of the DNA? Not so much mutation, but when we introduce things into the cell, we can change the expression of the DNA.
So what function would that be of changing the expression?
Hormones, for example, testosterone, estrogen, those can go into the nucleus and turn on certain things. So remember we said that lipids, they were nonpolar? Well, like steroid hormones are nonpolar. When we see how those functions, those will actually go through the membrane into the nucleus and turn certain things on. So to answer your question, can it go in there and cause mutation? Not really, but it can turn things on, turn things off. It can change the expression of DNA into RNA.
When that happens, does it not make the membrane bigger?
What, this? It does. Actually, yes, it will make it bigger, but there is a balancing act here. Because this is what we call, this fusing, there's also endocytosis and exocytosis. So certain processes, when something comes to the membrane, will take some of that membrane away. And other processes will add things to the membrane. So there's kind of a balancing act that there's an increase, and yes, it causes an increase in size, but then when we have something come into the cell, that causes a decrease in size. So there will be some balancing there. Okay, so now we have added proteins to our membrane, and then we can also go somewhere else. Here is the third one here. Here is our transport vesicle, and that can carry enzymes to another place inside the cell. Okay, so what can we do? We can release things outside the cell, we can integrate things on the outer cell membrane, and we can carry proteins somewhere else in the cell. Okay. All right. All right, new topic, mitochondria, another organelle. When you hear the word mitochondria, what function comes to mind? Energy what? Making energy, we'll get to that in my, we talk about metabolism. Let's break this habit. It doesn't really make energy. Okay, and I know when I was in high school, that's what I was told. It's like, it makes energy. It doesn't so much make energy, it converts energy into a more useful form for the cell. We'll get to this in metabolism, but what it does, and let me just use an analogy here. $100 bill. I give you a $100 bill. Remember it snowed a few months ago, right? I give you a $100 bill, I give you a stack of $100 bills, and you're locked in this building snowed in. Are that stack of $100 bills going to do any good? No. Can you use it in a vending machine? No. You go to most places around here, there's a sign, 20 bills or lower, right? You can't use a $100 bill. So, where am I going with that? $100 bill is not a very useful form of currency in many situations. Likewise, glucose in our cells is not a very useful form of energy unto itself, just glucose separately. So we have to have some type of mechanism to convert that glucose into ATP. Just like to make money useful, you have to convert it from a $100 bill to many $1 bills. So it's going to convert one form of chemical energy into another form of chemical energy. It doesn't make energy, it just converts it into a more useful form. Okay. Now, why do you want a $100 bill? Why is $100 bills good? It's easy to carry around. You're going to store money, right? If I want to store money, I'm going to store it in $100 bills. Because I can put 20 or 30 bills and I got a lot of money. Have you ever held like $1,000 in $1 bills? That's a lot of bills to keep track of. If you want to transport that around, is that great to transport? No, it's stupid. You're going to carry $1,000 in $1 bills. You're going to be carrying a big wad. Okay. Point is, sometimes money's good in $100 bills. Sometimes it's good in $1 bills. Sometimes energy is good in glucose, sometimes it's good in ATP. So where are you going to find mitochondria? You're going to find them in your cells, but you're going to, again, it goes back to the idea of a generalized image. Different cells have different amounts of mitochondria depending on their function. For example, muscle cells, you're going to see a lot of mitochondria with muscle cells because you need ATP to make a muscle work. You also need ATP to make a muscle relax, too. So you're going to see a lot of ATP production there. You're going to see a lot of mitochondria there. They're not placed at random. The mitochondria needs oxygen to work. You're going to see it closer to places where there's oxygen. Okay. All right. Any questions so far? We're good? Okay. Mitochondria structure, it has a dual membrane structure. A dual membrane structure. Okay? It has an outer membrane and an inner membrane. The inner membrane is where the reactions happen. Now the outer membrane, the inner membrane is highly folded. This orange, you see how it's folded like that? There's a reason for that. When we get to metabolism, we're going to see that the ATP production, there are certain enzymes and there are certain protein structures that are important for that. To maximize surface area, it's highly folded inside to maximize the area that we can have these ATP generating structures. For the purposes of this test, just know that the inner membrane is highly folded. We will get to why that is later on, but for right now, dual membrane structure, the inside is highly folded. But you will see this in a future test. It maximizes surface area. There are certain structures, ATP synthase, that's inside, and we want to maximize the area. More area, more of those structures we can have. So, and again, it goes back to its ATP production abilities. Okay. Okay, endosymbiosis theory. Where does the mitochondria come from? And this is one of those walks like a duck, talks like a duck type of ideas. We don't have a DeLorean. I hope that reference is not lost on anyone. No? Is it lost? Back to the Future, DeLorean goes back in time? All right, so we can't go back in time and tell you where the mitochondria comes from. But what we can do is what we do in biology. We look at what we see and we make the best guess possible. Okay, or we suggest a theory that fits those facts. And I don't want to say guess, that's not a really good way of saying it. We try to take the facts at hand and make the best, most rational explanation. And the most rational explanation as to where the mitochondria comes from is the endosymbiosis theory. Endosymbiosis theory goes like this. Back in the day, back in the day, Bill and Ted, not Bill and Ted, but Wayne's World reference there. Back in the day, what happened was there was a bacterium, we believe, or some prokaryotic organism that entered into the cell, and that prokaryotic organism took root and became what we would call the mitochondria today. Why do we believe that? Well, one, it has circular DNA. We call it plasmid DNA in bacteria. The mitochondria has this. In fact, this has been used. Has anyone heard of mtDNA or mitochondrial DNA? You can actually have your mtDNA sequenced, and you can use this to look at your genealogy on your mother's side. Typically, you're going to get not always, but most of the time, you get your mitochondrial DNA from your mother. So if you want to track your maternal lineage, you can actually use mitochondrial DNA for that. When we look at that DNA, it is circular, just like bacterial DNA is, like bacterial plasmids. We look at the ribosomes in this thing. Its ribosomes are very similar to bacterial ribosomes. In fact, when we treat certain people with antibiotics such as erythromycin, we see that it can actually damage things that are high in mitochondria, like the liver, because it's attacking that bacterial ribosome. Okay? It's similar in size to a bacterium. You look at the size of a mitochondria and you look at the size of something like a bacilli, they look pretty close to each other. And also it replicates independently. All these reasons we combined in and we think that the mitochondria comes from a bacterium. That's the endosymbiosis theory. Any questions? Okay. Well, no, actually, that's totally no. Take that thought and take it and flush down the toilet, man. No, actually, most bacteria are good for you. We would, if there was no bacteria, guess where we would be here? Nobody, it would be nothing. Okay, we're going to see bacteria have roles in ecology. We see that bacteria, if you're in your gut, okay, bacteria in there helping you. Okay, bacteria are your friends. Now, can bacteria be pathogens? You bet. Can some bacteria be bad? Yes. But if it was not for bacteria, there'd be no us. So, for the most part, bacteria are pretty good. I'm a microbiologist, so saying that term bad is like them is fighting words. All right. Okay. Another type of organelle, we're going to see these in plants, are things like chloroplasts. Now, we're going to really delve into chloroplasts later on. When we start talking about photosynthesis, we're going to be really concerned about what's going on in these chloroplasts. For now, we're just going to say that they're organelles, but you will see these in the future. Chloroplasts, what do chloroplasts do? What does photosynthesis do? Yes, well, again, it's going to convert light energy into ultimately ATP and glucose. Correct. Correct. It's going to convert light energy into a chemical form of energy. Okay. First, it's going to make ATP. Then it's going to take carbon dioxide and combine that and make your carbohydrates. All right. Questions? All right, so far so good. Central vacuole, we'll get to this later on. Also, when we talk about plants, inside of plants there is a large chamber in the center part, used as a storage place. Okay. Also, the pressure here keeps the cell somewhat enlarged. I believe this semester you'll see this when you look at the cells in your introductory biology labs. You will see this in lab. Okay. Quick question here about the Golgi body. You might even see the answer. Does a plant have a Golgi body in cells? Yes, it does. So plant cells do have Golgi bodies. And the reason is, do all plant cells do photosynthesis? Do all plant cells do photosynthesis? No, how about the cells in the root? How about the cells down the ground? Do those do photosynthesis? There's no light, right? They've got to convert carbohydrates into ATP. What are they going to use to do that? The mitochondria. Mitochondria. All right. Okay, we'll get to those. Lysozymes. Okay, eukaryotic cell walls. Many eukaryotic cell types do have cell walls. Fungus, they have a cell wall. Plants have cell walls. Fungal cells, they have cell walls. Animal cells do not. We do not have cell walls. Okay? But that doesn't mean all eukaryotic cells lack these cell walls. We do see them in eukaryotic organisms. And we also see them in our prokaryotic organisms as well. Okay. Questions, comments, thoughts? All right. And now we're going to talk about, we are multi-celled organisms, right? Right? So there has to be some way of connecting our cells together, right? If we are multicellular, don't you think that it's probably important how we connect the cells together? And it is. It is. These are our cell junctions. We're going to talk about 3 types. All right. It is in this order, tight, adhering, and then gap. So we're going to talk about each one of those separately. Okay. So the first one is a tight junction. Tight junctions are watertight bonds between cells. You know, think of caulking. You ever do caulking between your toilet and the floor? I know everyone's ever done caulking before. It's terrible work to do. But you've done it before, right? You're trying to make a watertight seal between those. Bless you. You're trying to make a watertight seal between those two, right? Well, tight junction is going to do the same thing for us. Why do we want these tight junctions? Why do we need them? Well, you start getting into the blood-brain barrier. Okay, really important when you start talking about how the brain gets nutrients and the physiology of the brain. We're going to see tight junctions there. Depending on the cell, there might be a lot of tight junctions. It just depends on what cells you're talking about. But these are essentially a watertight bond between cells. Okay. Another cell type is adhering junctions. Adhering junctions are like a spot weld. Adhering junctions are like a very strong connection. The emphasis here is strength. Not so much water tightness, but just physical strength. You know, some of our cells are under an intense amount of pressure and pull. What do you think? Think of some parts of the body. What type of cells do you think of are under a stress a lot of times? Muscles. Muscles. How about your bladder cells? Right. Your bladder fills with what? Urine. Okay. It's gonna expand. Right, and contract multiple times. So some cells need a very strong connection between their adjacent cells. That's where adhering junctions come into play. It's like bolts connecting them. It's like spot welds that connect cells. Another type of connection are gap junctions. We're going to see these, for example, when we start talking about the physiology of the heart. We're going to see these are used to transport, these are used to communicate between cells. So if you see the picture down here, we can use this as a means of sending signals between adjacent cells. Those are gap junctions. Now, which junctions do you see? Again, it goes back to the function of those cells. Oh, I'm sorry for this flickering. Okay. Any questions? All right. Next thing is the cytoskeleton. Very important part of a cell. The cytoskeleton. Now, do you remember we started talking about those pictures and I said, you know, there's a caveat that those are the best pictures. They're trying to convey an image. What those pictures did not show was the cytoskeleton. The cytoskeleton is this proteinaceous network inside of a cell. Lots of functions with this one. Okay. So just to give you a couple examples here. It determines the cell shape. Organizes the contents. We'll show how that does with motion in a minute. Moves chromosomes during cell division. When we start talking about mitosis and meiosis, what does mitosis and meiosis do? Splits the cell. Specifically, what does it split? What part of the cell? You're right. The nucleus. Correct. Correct. We're gonna have DNA in the nucleus, and we're gonna split and pull that DNA apart. Well, our cytoskeleton is going to be important in that pulling apart of DNA. Specifically microtubules. So we'll talk about specifics in a minute here. But there's lots of functions in the cell that our cytoskeleton is responsible for. When we release things like vesicles, right? We release a vesicle from one area and want it to go to another area. Does it float along like a balloon, and does the cell hope, I'm going to release a vesicle over here and I hope it floats over there? No, it doesn't do that. We have a cytoskeleton network that we can direct vesicles along from one area to another area. So there's a lot of functions here. We're going to talk about some of those functions moving forward. All right. So the three parts to this is a microfilament, an intermediate filament, and a microtubule. Let's just use some logic here. What thing do you think of when you think of a filament? A what? Light bulb. Light bulb, exactly. I think of a light bulb. When you look at a light bulb and look it up in the air, what do you think about that filament? What comes to mind? Thinness or thickness? Thinness, right? You look at a light bulb filament, it's a very thin little wire inside the light bulb. So let's think of the idea of filament as being very thin. Okay, how about filament? Tubule. When you think of the word tube, what do you think of? Big old thing. I think of something like a PVC tube. I think of something like a tube like at a water park. I think of something big. Don't you kind of think of something big when you hear the word tube? Well, a microtubule is going to be the largest of these filaments. And when I mean largest, I'm referring to the diameter. See how this is the thinnest, thicker, and the thickest? That's the size that I'm referring to. Okay. So microfilament and then intermediate. How are you going to remember the intermediate? It's in the middle. All right. All right, so let's start with the microfilament. Microfilaments are made of actin. Actin. Where have you heard the term actin? Actin. Where have you heard actin? Muscle physiology. Where have you heard of that? That's really important in muscle physiology. You'll probably discuss that. You'll discuss that in the second semester, how a muscle works. But we have actin and we have myosin in a muscle. Actin is a very important part of how your muscle works. Okay? Part of the cytoskeleton. Also responsible for helping that cell separate. We'll talk about that later on too. Traktile ring. Okay. Now, how does actin work? Actin will polymerize spontaneously. So I got a little video here, and we have actin. Actin are made of monomers, and those monomers come together, and they polymerize, and they form a larger structure, a polymer. Okay? It happens pretty fast. Now, it looks like it's happening really fast here, but in actuality, it's much faster than what that little video shows you. So actin molecules will polymerize fairly fast. Next are intermediate filaments. Intermediate filaments are between your microfilaments and your microtubules. The emphasis here is mechanical strength. Scaffolding. These are very strong. They don't depolymerize like the actin we just saw. At least not to that level. Okay, and what's the last one we're going to talk about? What's the last one? The largest one. Microtubules. Microtubules, these are made of something called tubulin. And tubulin will polymerize or depolymerize at the end. So what we're showing here, if you see that on the corner left, we have a tube. It's literally like a tube. And they will come together and polymerize. Microtubules come together and they polymerize. For them to cause motion, they will depolymerize at one end, and as they depolymerize, it will pull on that tube. So that tube depolymerizes. As it depolymerizes, the rest of the tube pulls along. Okay, it will depolymerize from the end, and as it depolymerizes, it's pulling the rest of the tube the direction is depolymerizing. When it depolymerizes on one end, it pulls the other end. So for example, if we're trying to pull DNA apart, one end will depolymerize, and as that end depolymerizes, the other end being attached to DNA will pull and separate that DNA in mitosis. Now, where do we see these microtubules working? One, we separate DNA in mitosis and meiosis. We're going to depolymerize on an end, and as it depolymerizes, it pulls that tubule. Another way, and we'll get to this in a second, is a pathway for vesicle transport. Remember I said we don't release vesicles like balloons and hope for the best? What's going to happen is we have a motor protein attached to some vesicles, and it will use the microtubules as a pathway, a superhighway, a causeway, a thoroughfare, whatever. And it will move our vesicles down from one area to another area. Again, it's not like balloons being let out and hoping for the best. Okay. Cell movement, cilia and flagella. Where are we going to see flagella in humans? Where do we see flagella in humans? For movement, causes movement. What human part has this? Only one gender has it. Sperm cell, exactly. We have a flagellum on the sperm cell. It's called a 9+2 arrangement. But this moving will cause the sperm cell to move. Okay. All right, but there's lots of examples here we'll get to later on. But it causes movement. All right. Another one here, this is a mitochondria. Those lines you see are microtubules. And what's going to happen is, again, we're helping move organelles. You know, they're not static entities. They're moving from one area to another area. Okay. Now, has anyone ever heard the drug Taxol? No? Parataxol? Okay. This helps block the depolymerization of microtubules. And when we depolymerize a microtubule, what happens to the rest of the microtubule? It pulls, right? So, for example, this is my microtubule, right? And it's connected to DNA, and here's my end that I'm depolymerizing. As I depolymerize this end, what happens? It does what? It pulls the other thing. Exactly. That's basically how DNA separates. So that depolymerization causes a pulling effect. So if we could somehow block that depolymerization, guess what we could do? What is cancer? Uncontrolled cellular replication, right? DNA is always being separated, made and separated. So here we're going to block the separation of DNA by blocking the depolymerization of microtubules. So anyhow, that wraps us up for that material. Is there any questions?